It's my pleasure to introduce Dr. Cohen. Dr. Cohen is a medical oncologist at the University of Pennsylvania in Philadelphia. He's a graduate of Harvard Medical School, completed internal medicine and hematology training at Mount Sinai Hospital, followed by research fellowships at the Memorial Sloan Kettering Cancer Center and the National Institutes of Health, and a medical oncology fellowship at the National Cancer Institute. He's an active investigator on several first-in-humans clinical trials with research interests that focus on evaluation of novel therapies, including monoclonal antibodies, immune therapies, and small molecule cell signaling pathway inhibitors. Dr. Cohen, you are on.
Thank you very much. Thank you to the organizers of this meeting for inviting me once again to talk about CAR T-cell therapy for medullary thyroid cancer. The host of the meeting is going to advance the slides for me. And I'm also speaking on behalf of my co-investigator, Dr. Siegel. Dr. Siegel is at his own professional conference today, this weekend, and thus is not able to present a portion of this deeply personal story that I'm going to share with you on his behalf. Next slide, please. Dr. Siegel's friendship with Dr. Carlos Barbas inspired the science behind this experimental treatment. Dr. Barbas died of medullary thyroid cancer in 2014. He was a PhD scientist with a deep interest in something called phage display, which is a way of generating antibodies to just about anything. And Dr. Barbas was one of the pioneers in this particular field. And although dying in 2014, thus he did not live long enough to see the Nobel Prize in Medicine or Chemistry awarded for this particular set of discoveries. It was awarded to 3 investigators from across the world. Dr. Barbas developed thyroid cancer and unfortunately had a particularly virulent version of this cancer and died within 6 months of diagnosis. Next slide, please. I'm just going to walk through a little bit of the science behind the CAR T cells and how they were developed. And they have everything to do with Carlos's medullary thyroid cancer and Carlos's DNA in that medullary thyroid cancer. On the left-hand slide, I'm showing you a conventional pathology slide showing follicular cells which make thyroid hormone. And that has nothing to do with medullary thyroid cancer. Medullary thyroid cancer arises from cells that are in the thyroid gland. They're called parafollicular cells, and they make calcitonin, and they are shown with the arrows in the upper right-hand corner of this slide. Next slide, please. And this is an illustration of the RET gene. The RET gene underlies the development of medullary thyroid cancer in many, but not all cases of malignancy. And RET is a tyrosine kinase. It is a signaling molecule. It spans the cell membrane. And when activated, it sends signals into the center of the cell on the bottom part of that diagram. And those in turn lead to activation of a number of molecules that cause cells to divide, allow cancer cells to survive, as well as move around, metastasize, and so forth. RET is a normal gene, but when mutated, it is always on. And so it's like leaving a switch on in your home. It's not a good thing. And the cells proliferate indiscriminately and metastasize. Next slide. Carlos had a mutation in RET, and it's shown in the oval there. Go back, please. And it's shown in the oval there. That is his particular mutation in the RET gene, which was in fact the root cause of his medullary thyroid cancer. He did not, however, respond to RET inhibitor therapy. Next slide, please. Dr. Siegel wondered, could he use Carlos's breakthrough technique to make a CAR T-cell therapy for his friend? Advance, please. And so he wanted to use Carlos's scientific method to identify a target for immunotherapy. And he wasn't sure when he started what he would find. Would the target be Carlos specific, something absolutely unique to Dr. Barbas's tumor? Or would it be more characteristic of medullary thyroid cancer cells generally? Or would it be some kind of parafollicular or C-cell specific target? Advance, please. And if he could find a target, he could then make an antibody against that target using Carlos's breakthrough techniques. Next, advance, please. And use that particular antibody to fashion a CAR T-cell in order to target and kill medullary thyroid cancer cells. Next slide. So they identified some potential targets by analyzing Carlos's medullary thyroid cancer. Next slide, please, or advance, please. And they identified a very large number of targets, and not all of them are suitable for a therapeutic. Advance. And this is a summary of some of those genes in the left-hand panel. Advance, please. And they settled on GFR alpha 4. And I'll tell you why in a moment they settled on GFR alpha 4. But the bottom line is that this particular target was overexpressed in medullary thyroid cancer cells, including Carlos's, but not expressed barely at all in normal cells, normal tissues in the body. Advance, please. What is GFR alpha 4? Advance, please. It's specific to T cells, and what it stands for, or what it's a member of, is a glial-derived neurotrophic factor receptor family. You don't need to remember that. There are 4 members of the family. GFR alpha 4 is one of the 4 members that's restricted to C cells. Those are the cells that go rogue in medullary thyroid cancer. And it actually has nothing to do with RET mutations, and its function is unknown. Advance, please. But one nice thing about it is that its expression, and these are some scientific slides, these are blots, just showing in the very top panel, you can see those little white bars showing that GFR alpha 4 is expressed in the thyroid gland, and then you have a whole host of other normal tissues from the human body in which it is not expressed. Next slide. And this is showing expression on slices of tissue stained in the pathology lab, showing that GFR alpha 4 is found in the normal thyroid. Remember that slide I showed you a moment ago with all the pink stuff, showing the parafollicular cells. There they are with those little arrows. It's expressed there. MTC1 is Carlos's tumor, and you can't see the brown stains clearly at a distance, but they are there. So it's overexpressed in his tumor. It's overexpressed in another tumor. And then there's also a cell line called TT in which it's very strongly expressed. Advance, please. And this is just a narrative depiction of the fact that it's expressed in medullary thyroid cancer cells, the TT cell line that I told you about a moment ago, the normal thyroid gland, but it's not expressed in any of these other tissues, which is very convenient because it gives specificity. You can attack this particular target without causing collateral damage to other parts of the human body. Next slide. And so Dr. Siegel wanted to know next, could he actually make a CAR T-cell therapy for Carlos using this particular target? So we've identified a target. We are now going to use those methods that Dr. Barbas had taught Dr. Siegel years earlier. Dr. Siegel made an antibody using Carlos's techniques directed against GFR alpha 4 an antibody with the name P410. Now, the next step in the process is to make a CAR T cell using that antibody to target medullary thyroid cancer cells. Next slide. What is a CAR? A CAR is a chimeric antigen receptor T cell. A chimera in Greek mythology is an animal that's composed of the head of a lion, the body of a goat, and the tail of a snake. Advance, please. And so what they did is, using the phage display technology that Dr. Barbas invented, they found an anti-GFR alpha 4 antibody. And what they're going to do now is fashion a new T cell receptor that can be used to genetically alter T cells. So if you look in the lower part of this slide, you see a T cell depicted there. The membrane is the yellow stuff at the top. These are receptors that recognize foreign proteins, for example, COVID virus, and help the body get rid of it. And what they're going to do is make an artificial T cell receptor using this anti-GFR alpha 4 antibody that was discovered using Dr. Barbas's technology. And so they're going to swap in that blue turquoise element there at the top, and they're going to then fuse it to the rest of the T cell receptor. Advance, please. And so what you end up with is a chimera. You've got the head, which is an antibody, the body of the protein, which is called CD8 or CD28. This is very important for helping T cells recognize and destroy their targets. And you've got the tail of the T cell receptor complex, which has a complicated name, but that's also part of the mechanism whereby these receptors on T cells recognize foreign substances like cancer cells, like COVID virus, and help the body get rid of them. Advance, please. So how do we actually make CAR T cells? They are made by harvesting blood from patients. That's called leukapheresis. So there's some numbers here, 1, 2, 3 through 9. So step number 1 is leukapheresis, harvesting the blood, and that is then put through a machine that helps us purify white blood cells, and the rest of the cells, the red cells and so forth, are given back to the patient. These white cells are then concentrated using those red beads on the lower right-hand corner, and they are artificially transduced with a virus that contains that made-up T cell receptor construct that I showed you in the previous slide. So these cells are being systematically altered so that they now possess this T cell receptor that recognizes medullary thyroid cancer. The cells are then stimulated and grown up in what's called a bioreactor over a couple of weeks, so that you turn a few cells into millions and millions and millions of cells. These cells are then concentrated, washed, and put through some quality control measures on the left-hand side, and you end up with the final T cell product, which is in a tiny little plastic bag that can then be infused back into the patient. Advance, please. And this is just showing this process of leukapheresis, which is a little bit like donating blood. That machine that purifies the T cells is standing in front of the technician at the University of Pennsylvania. Advance, please. And this is an animated slide. So I've told you a moment ago, and this is just different ways of showing the exact same thing, that we have this artificial, man-made T cell receptor that contains this head, which recognizes the GFR alpha 4, that's at the top in red. And then it goes through the cell membrane, and then it has these other domains that are critical for helping the T cell recognize the target, GFR alpha 4, and then activate in order to do its job, which is to kill. And so I'm just going to animate this by asking my helper here to advance. So here you've now introduced this artificial man-made T cell receptor that's illustrated to the left. And now it's sitting in the membrane of the T cell. Advance, please. This particular construct is designed to recognize medullary thyroid cancer cells and then activate the T cell in order to do its business and kill them. With these CAR T cells, we are learning that they can be infused once and are capable of living in and dividing in the body and persisting in the body, sometimes for years. And in the case of some of the patients that have been treated with leukemia, for more than 10 years. Advance, please. How did the use of CAR T cells for cancer start? Didn't start that long ago, about 13 years. Next slide. And this was in August of 2011, the publications of the initial use of chimeric antigen receptor modified T cells in chronic lymphoid leukemia. And it is in blood cancers like leukemia that this field is furthest along. What I'm talking about today is CAR T cells for solid tumors, an area that frankly is immature right now. We are learning our way, finding our way, just as scientists did 12, 13 years ago with CAR T cells in leukemias and lymphomas. Advance, please. This is a picture of CD19 CAR T cells, which is now an FDA-approved product, killing a leukemia cell. The CAR T cell is in the middle, and the leukemia cell is in the upper right. The CAR T cells attach themselves to the leukemia cell, and then the T cells, once activated, inject all kinds of poisons into the leukemia cells, killing them. Next slide. The very first patients treated this way had CLL, chronic lymphocytic leukemia. This is a picture of one of them. These are very public individuals who have spoken to various news organizations and patient support groups about their experience. Next slide. This is just an example of what happened in this particular patient whose bone marrow was completely filled with CLL cells. And by 30 days after being treated with the CAR T cells, all of the abnormal malignant B cells had been killed. Next slide. This is actually patient number 2. And patient number 2 is standing next to Dr. Siegel. Dr. Siegel, of course, is the scientist who is behind this whole story, and he was Dr. Barbas's friend. Next slide. And here are actually both of the first patients, number 1 and patients number 2, some 10 years later. Next slide. And this is a very important article that came out not that long ago, demonstrating that these CAR T cells could be found, not in very large numbers, but they could be found in the blood of patients who had been treated almost 12 years earlier and gone into remission from their CLL. Next slide. CAR T cells are also being used successfully to treat pediatric ALL, acute lymphoblastic leukemia. This is patient number 1, another public figure, Emily Whitehead. Next slide. Emily recently matriculated at Penn. She is a member of the class of 27. Next slide. There are a number of CAR T cells in development. So with all of the successes in lymphoma and leukemia, it goes without saying that scientists and medical oncologists are keenly interested in developing analogous CAR T cells for all of the malignancies that plague humans, ranging from pancreas to gastric to brain tumors, ovarian cancer, bladder cancer, etc. And it is in this particular space, the so-called solid tumor space, that the CAR T cells for medullary thyroid cancer fit. Next slide. So getting back to Carlos and the GFR alpha 4 directed CAR Ts. Advance, please. So this has been published, the development of these cells and their use in preclinical experiments in dishes and in mice to establish that the technology might have promise if put into humans. And you will notice the publication date here, January 2021, so it's not that long ago. Advance, please. I'm going to show you some graphs. I'm going to try to explain them in lay terms. But what is being done here is that these TT cells, which is a medullary thyroid cancer cell line that I've shown you now a couple of times, is being exposed to these engineered CAR T cells in a laboratory experiment, and showing that as the number of CAR T cells is increased, cells are killed. Cells are actually killed even at the lowest level of CAR T cells. The lines at the very bottom are dummy CAR T cells that do not have this manufactured T cell receptor directed against GFR alpha 4, whereas the CAR T cells are killing tumors in the upper graph, the one that goes upward. Those are the armed, if you will, CAR T cells armed with this novel T cell receptor that has been transduced or introduced into these T cells. Next slide. You can also show that these cells can kill medullary thyroid cancer cells in a mouse model. Another scientific experiment. Advance, please. So mice are injected with these TT cells. The cells are allowed to grow in the mice. The mice are then infused with these CAR T cells in the laboratory. Next slide. And I'm just showing you here that these mice whose tumors are labeled with a fluorescent tag. And I'll show you in a moment what that looks like. So you can actually see the CAR T cell tumors, or you can see, excuse me, the medullary thyroid cancer tumors in the mice. And on the left, and you might as well advance so you can see what these pictures look like. And so here are the mice. And the mice have these tumors growing in their flanks, and you can see that they are colored. They are red, for example. And you can see that if the mice are infused with the CAR T cells on the left-hand side, you can watch the photofluorescence drop off as the tumors are killed. You can also see in the middle panel B that tumor volumes are decreasing when the mice are infused with these GFR alpha 4 CAR T cells. And perhaps most vividly at all, if you compare the mice on the left-hand part of the slide on the far right, who did not receive the GFR alpha 4 CAR T cells, but instead received the dummy cells, versus the mice that received the actual live CAR T cells with the GFR alpha 4 T cell receptor, you can see the fluorescence in their flanks go away. This is a very important proof of concept. Next slide. So on that basis, an IND application was submitted to the US Food and Drug Administration, providing the Food and Drug Administration with all of the experiments that established that the CAR T cells were active, at least preclinically, and that they were also likely to be safe. And this clinical trial opened. This is the name of the clinical trial. The NCT number is on the far right, and we are actively recruiting to this study. Next slide. I want to summarize the study for you, and I'm intentionally leaving time for questions at the end. So I've got about 20 slides to go. Next slide. So again, just what are we doing here? This is another illustration of the process of producing the CAR T cells, which we are doing and have done in the clinical trial, putting patients through this leukapheresis procedure to harvest white blood cells. The white blood cells are concentrated in a laboratory and are transduced. That's the technical term for the gene transfer to put in this artificial T cell receptor. The transduced or altered cells are then expanded. They are then harvested after being expanded for a couple of weeks, and now you have several billion cells. The patients are then put through something that's called lymphodepleting chemotherapy. Lymphodepleting chemotherapy is a chemotherapy over 3 days that is designed to eliminate some of the normal T cells in the body. And the reason that that's being done is to create space for these modified T cells in which these T cells can expand because that's very important for their cancer-killing properties. And the lymphodepleting chemotherapy is an important part of CAR T cell therapy. It's something that really cannot be eliminated at this particular point in time. The chemotherapy does not affect the medullary thyroid cancer at all, but it does create space for the modified T cells to live, hopefully divide and prosper for a while in the human body. Next slide. And so just to remind you once again, and this is an animated slide, as before, it's a repetition, just showing you the construct on the left that was transduced into the T cells. Advance, please. Showing that these T cells from the patient are now armed with this GFR alpha 4 CAR construct. Advance. And the theory is that these modified T cells will identify, recognize, and subsequently kill the medullary thyroid cancer cells. Next slide. I've told you that GFR alpha 4 is thought to be a good tumor target, and I want to tell you why and remind you of some things I've already said. GFR alpha 4 is expressed on the surface of all medullary thyroid cancers that have been tested in the laboratory. And GFR alpha 4 expression is limited to the parafollicular cells. Those are the cells that make calcitonin and that turn into medullary thyroid cancer cells. And in that respect, the GFR alpha 4 is very analogous to CD19. So CD19 is the target of the CAR cells that were used to treat the patients with leukemia, including Emily Whitehead. The nice thing about CD19 is that it's only found on B cells, normal B cells and malignant B cells, and GFR alpha 4 is only found on the cells that become medullary thyroid cancer. And that creates an enormous potential window of opportunity to kill cancer cells without collateral damage. Now, one expected effect of the GFR alpha 4 CAR T cells is that they will eliminate parafollicular cells that make calcitonin. And that means that you won't be able to produce calcitonin anymore, which is probably irrelevant for most people with medullary thyroid cancer in whom the thyroid gland has already been removed. But it turns out that mice and people can actually live normal lives without cells that make calcitonin. And indeed, the function of calcitonin in the human body remains a bit of a mystery. Next slide. So the study objectives are to evaluate the safety of these cells, and importantly, to evaluate the manufacturing, the production of these cells, which involves lots of steps, as you've seen now on a couple of different slides, and also to describe preliminary anti-cancer activity. Next slide. We also want to determine the bioactivity of the CAR T cells in blood. And specifically, we want to know, can we find the CAR T cells in the blood of our patients? How long do they stay there? If we can safely perform biopsies because they are feasible and not near things like major blood vessels, can we see if the CAR T cells actually get into tumors? And then we are measuring a range of immune and inflammatory substances in the blood before and after the CAR T infusions, in part because those substances mediate some of the potential side effects of CAR T cells, as I'll tell you in a moment. Next slide. This is a summary of the so-called eligibility criteria for the study. So I'm just going to give a top-line summary. It's adults with the disease who have experienced worsening of their disease after at least one tyrosine kinase, TKI or MKI therapy, or the patient was intolerant of or declined such therapy. I didn't put in this slide, but it's very important. This is not a RET-directed therapy. This particular therapy is RET agnostic. So it's suitable for patients with RET mutations or lacking RET mutations. To get in the trial, you have to have adequate organ function, kidney, liver, heart, and lung. Performance status just refers to your ability to execute activities of daily living. 0 is no symptoms, 1 is some symptoms, but you're still functional and, for example, could work at a desk job. You've recovered from the important side effects of prior therapies. You have to have something that we can evaluate on a scan. so that we can tell whether there's been clinical benefit or not. And subjects of reproductive potential must agree to use birth control methods. Next slide. People who cannot be in this study are those with active infections or clinically important heart disease, including clinically important arrhythmias. This is an immunotherapy, and therefore we don't want people on high doses of steroids, because high doses of steroids, such as prednisone, will undermine the effectiveness of any immune therapy. An increasing number of patients are receiving these so-called immune checkpoint inhibitors, drugs such as pembrolizumab or Keytruda, and so we ask that people not have received such a therapy within the preceding 2 months. Again, this is an immunotherapy, so we are a little bit concerned about activating autoimmune diseases. So we don't want people in the trial with active autoimmune disease, which means diseases such as rheumatoid arthritis, systemic lupus, ulcerative colitis, Crohn's disease, multiple sclerosis, and the like. And although medullary thyroid cancer does not commonly go into the CNS, we are excluding patients with active CNS disease, in part because of the concern that the CAR T cells would have a great deal of difficulty actually getting into the central nervous system. Next slide. This trial has 3 dose levels. These dose levels are very similar, if not identical, to those used by our university and others in studies evaluating other solid tumor CARs. The CARs that are the furthest along are directed against a substance called mesothelin, which is expressed in mesothelioma, ovarian and pancreatic cancer. And we are modeling this particular study after those other solid tumor studies. And of course, that is done in part in collaboration with the US Food and Drug Administration, which regulates the performance of these highly experimental clinical trials. I told you a little bit about lymphodepleting chemotherapy a moment ago. So that is given over a 3-day period, and the last day is 3 days plus or minus 1 day prior to the actual infusion of the CAR T cells. There are a variety of lymphodepleting regimens that can be used. The one that we are using, because it's the one that we have the most familiarity with, involves 2 drugs called fludarabine and cyclophosphamide. And then following the lymphodepleting chemotherapy, our patients get a single dose of their manufactured CAR T cells via intravenous infusion over a few minutes. Next slide. The study consists of 5 phases. We meet the patient. If the patient is eligible and signs consent, they then go through the apheresis process where they donate their white blood cells so that we can bring them to the lab and manufacture the cells, a process that takes a couple of weeks. The treatment phase includes the lymphodepleting chemotherapy that I told you about a moment ago, as well as the infusion of the CAR T cells, followed by short-term as well as long-term follow-up. And the primary follow-up, the short-term follow-up, is pretty intense because we want to see our patients weekly for 4 weeks after the infusion and then less often thereafter. Again, this is modeled after all of the other CAR T cell experiments that are being done in the United States, as well as the CAR T cell protocols that were the basis of the successful registration of the CD19 CAR T cells for the treatment of lymphoma and leukemia. Next slide. We do have a retreatment provision in that subjects who have had some clinical benefit can get retreatment. The retreatment will be with leftover CAR T cells that are in a freezer, and this can occur at the physician's discretion in collaboration with their patient. Next slide. Side effects are very important with this class of therapeutics. And right now, we don't really know what the side effects are going to be, but we do have some concerns. Next slide. I've shown you something similar to this before, and the concern is that if you make a solid tumor CAR T cell and you're targeting, for example, medullary thyroid cancer or something like pancreatic cancer, how specific is your target? Is your target restricted to the medullary thyroid cancer cell, or is the target more broadly distributed in the human body with perhaps expression on the heart or the nervous system or some other critical structure? This was looked at in some detail, both in human tissue specimens that are commercially available, as well as in monkeys. And this is a monkey experiment showing you that this particular target is not expressed in human tissues other than thyroid, other than medullary thyroid cancer. And the monkey, the cynomolgus macaque, is a reasonable model for this purpose because the monkey expresses this particular target in thyroid and does not express this target in other cells. So you can do experiments in monkeys and show that the CAR T cells will not result in catastrophic damage to the animals. Next slide. The other issue with CAR T cells that you have to be worried about is that they have a unique mechanism of action of activating the immune system to kill cancer cells very quickly. So you're injecting a large number of T cells, they are armed, and they are trying to kill these cancer cells. And if they kill the cancer cells very quickly, you can get something called cytokine release syndrome. So this is a class effect and is a theoretical possibility for any CAR T cells, and it certainly has been observed in all of the patients who've been treated with leukemia. And cytokine release syndrome is the instantaneous release of immune mediators, which cause fever, chills, low blood pressure, and even organ damage from an overwhelming immune response. Think about, for example, what happened to some of the patients with COVID. They experienced an overwhelming immune response and as a result had organ damage. There's also something called neurological toxicity, which is another concern and has been observed in CAR T cells used in liquid malignancies. And this can range from a little bit of confusion for a couple of hours to a coma. Fortunately, it is reversible, but it can take some time to reverse. And remember, I told you that these CAR T cells will eliminate any leftover parafollicular cells that make calcitonin. The long-term side effects of CAR T cells are simply unknown. Remember the publication date of the original use of these cells in 2011. So we are still learning, and we are definitely very much in a learning phase for the so-called solid tumor CARs, of which the GFR alpha 4 CAR T cells are representative. Next slide. This is just a summary of cytokine release syndrome, which I told you a little bit about. It can be mild, in which case it can be managed with Tylenol, but higher grades of CRS, which we certainly have seen in the leukemia patients, can require ICU admission, lots of fluids, drugs to raise blood pressure, intubation, as well as these anti-cytokine therapies such as tocilizumab or cetuximab. These are FDA-approved therapies. They're not approved for CAR T cells necessarily, but they can antagonize this overwhelming immune response and shut it down pretty quickly if the patient is getting into real trouble. Next slide. University of Pennsylvania has been instrumental in developing the CRS management algorithms that have been published. I'm not going to walk you through this, just to say that there's a very well-trodden roadmap for managing or identifying this toxicity as well as getting patients through it. Next slide. The neurological toxicity is by an unknown mechanism, and it occurs later than the CRS that I just told you about, generally not life-threatening, although it can be, and it is reversible. And what we would do were we to observe this in our patients on this clinical trial is consult neurology because they have a lot of experience managing this particular side effect. Next slide. And these are some potential side effects that have not been observed, but one has to mention them for completeness. So we are using a virus to put this artificial T cell receptor construct into these T cells. And there's always the possibility that this lentivirus could change while you're using it and become capable of replicating itself and regenerating itself. This is highly unlikely, but it's monitored. It is also possible that the T cells that you've injected into the patient might go crazy and proliferate wildly. And if that were to occur, we would treat people with steroids. Steroids can kill T cells. And there's always the possibility with this kind of genetic alteration of cells using these using these lentiviruses, that you could cause cancer by actually modifying the patient's normal DNA. That has not occurred, but it remains a hypothetical consideration whenever you're doing a type of gene therapy, which is what the generation of these CAR T cells involves. We have treated 2 patients on the study so far. You can go to the next slide. We have treated 2 patients on the study so far. We have learned some incredibly important things from those 2 patients. We have learned that we can consistently make very high quality GFR alpha 4 directed T cells in very large numbers, certainly adequate for therapy. We have also learned that we are not observing any of the feared side effects from these CAR T cells, namely targeting of normal tissues in the body. So that has not occurred, and that fits with the preclinical data that were submitted to the US FDA, assuring the FDA that this probably would not occur. We have not observed any therapeutic benefit at this time. That is to say, we have not seen any tumor shrinkage, and we have not seen any drops, for example, in calcitonin. The patients that we are treating have very advanced disease, and so that may be one of the issues with not observing therapeutic benefit. In any case, we continue to actively search for patients for this trial. This is fortunately a very rare disease, and fortunately, many patients with this rare disease are doing well on their TKIs, their MKIs for very long periods of time. But we are looking for patients where the effect, the benefit of MKIs and TKIs has run out, and are especially keen to enroll patients lacking RET mutations who have a paucity of useful therapeutics at their disposal. So I have intentionally left time to answer questions. My understanding is that ThyCa is managing the chat, and I will hear your questions and answer them, and I encourage you to contact me at the information that's listed on this slide. I thank you very much for your attention.
So we have questions online as well as here in the room, I'm sure. Anybody in the room want to start? Well, we'll start with online questions. Nobody in the room is raising a hand. So first question, Dr. Cohen, thoughts on funding for research in the future? Will pharmaceutical companies be interested in treatments like this, or do you feel they prefer chronic disease treatments?
I think that they will be interested. I know that they are interested. This particular trial is not funded by a pharmaceutical company. It is funded by philanthropy. So it is funded by a generous donation to Dr. Siegel's lab from a donor, a grateful patient at Penn Medicine. I've no doubt that were we to develop a successful GFR alpha 4, that there would be some interest from pharma. As you know, there are a number of very rare diseases that are being treated with these exotic therapies. The price tag is quite high, but if the drug actually works, and it is our obligation to show that, and we're only giving a single infusion, there are certainly examples in the real world. Spinal muscular atrophy stands out as one example. There's a form of blindness that is treated with a gene therapy. And so one shot, you might charge $500,000 to $1 million, but it only happens once and therefore becomes cost effective if the treatment itself really is effective. So I think there is interest. Obviously, there's a lot of politics involved here, and I don't really want to get into that, but I think there is interest in pharmaceuticals because this represents the future of medical oncology.
How long after the introduction of the engineered cells are participants expected to avoid chemo drugs?
28 days. So there is a dose limiting toxicity observation period. And the practical question that you're asking is, what happens to my MKI if I go on to this trial? So you would need to stop the MKI a week or two prior to undergoing the lymphodepleting chemotherapy. And so you're off of it for 2 weeks prior to the CAR T cell infusion. Then we ask that you also be off of it for 28 days following the CAR T cell infusion so that we can observe you for side effects related to the CAR Ts uncontaminated by overlapping side effects or non-overlapping side effects from the MKI. And then as soon as that 28-day observation period is finished, you can go back onto the MKI. And I'm using the word MKI or TKI because in general, medullary thyroid cancer does not respond to and is not treated with conventional chemotherapy as that term is understood.
Is it possible to reproduce MTC in mice to test?
Yes, so one of the slides that I showed you in the lead up to the clinical trial were the proof of concept trials in mice. Those are published in that molecular paper in January of 2021. Dr. Siegel is one of the co-authors, and you can see some of the graphs that I showed you as well as additional demonstrating proof of concept. So for the agency, what you need to do in order to perform a trial like this is demonstrate that the drug, in this case, the engineered T cells, are able to kill medullary thyroid cancer cell lines in a dish, but also create some animal models, both for safety as well as for proof of concept. And so if you think back to those mice with the colored circles in their flanks, those are the experiments that I was referring to in mice.
There's one more online question. Did the engineered cells expand sufficiently and persist a long time in those people similar to the blood cancer patients?
They did not. So they were detectable, and they were detectable for about a month. Now, are there any CAR T cells left at very low numbers? That is possible, and that's what you see in those long-term survivors with CLL, that the patients have very, very small numbers of CAR T cells in their body. Presumably, those CAR T cells can be activated and will then expand in response for the CLL patients, for example, to a relapse. So we did see the CAR T cells in the blood in very good numbers, and as I said, for about a month. We did not see persistence at high levels for a long period of time, and we don't understand the reason for that, bearing in mind that this is based upon 2 patients, and we've got a ways to go in order to more fully describe the levels of and the persistence of the CAR T cells in human blood. We were pleased just to see them, that they managed to survive and that they were present for a couple of weeks after infusion.
Dr. Cohen, we do have a question in the room, so I'm going to bring it to the live audience for a few.
Okay.
Dr. Cohen, since calcitonin is one of the main tumor markers to measure status of disease, if CAR T cell therapy eliminates the cells that produce calcitonin, do we lose completely the ability to utilize calcitonin to continue to measure disease, or does the ability to measure calcitonin at some point return?
Well, the best possible outcome, the dream, is to actually eliminate calcitonin once and for all, because the only cells in a patient with medullary thyroid cancer who has had a thyroidectomy that are producing calcitonin are the cancer cells. And so if we kill the cancer cells, and that is the goal, and the patient has had their thyroid removed as part of their therapy, mission accomplished. And the calcitonin is gone, and we hope to never see it again. I hope that answers your question.
Are there any other questions?
So the 2 patients you had on the clinical trial, did you biopsy any of, or do a fine needle biopsy or anything of the tumors to see if the white cells were even in the tumors, or?
So we did try. The first patient had a lymph node biopsy, but the amount of material that was obtained from a neck node was really too small to determine whether the CAR T cells were actually in the tumor cells. We could certainly see the evidence of the CAR T cells in the biopsy, because we get some liquid, we rinse the needle, and we could see the CAR T cell DNA in the biopsy, but we didn't have any spatial information, which is really what you need from a core biopsy in order to see the relationship of the engineered foreign T cells and the tumor. The second patient was ready to get a biopsy, but he has a needle phobia, and he was trying to be a good sport in being willing to undergo a biopsy, but he wanted to be put completely asleep for that, and the risks of that exceed the benefit, and so we forewent the biopsy in that particular case. The biopsy is critical information because one of the issues with solid tumor CAR T cells generally, and it's certainly relevant to this particular protocol, is are the CAR T cells getting into the tumors? If you think about white blood cells, think about Emily Whitehead's white blood cells circulating throughout her body. They are readily accessible to a CAR T cell that's infused into your vein. So the CAR T cell doesn't have to do very much in order to see the leukemia cells, identify them, attach to them, and kill them. With solid tumors, again, think about medullary thyroid cancer, pancreatic cancer, what have you. There's a structure to the tumor. It's like an organ. And the CAR T cells have to somehow recognize and penetrate to some extent into that fake organ, that abnormal organ, in order to kill the tumor cells. So this is an ongoing challenge. And I know that people in the Carl June lab and others are actually working on mechanisms to try to disrupt any of the barriers that might exist in these tumors to facilitate entry of the CAR T cells into the tumors. But the biopsies are absolutely critical if they can be obtained safely in order to see whether we have succeeded in getting the CAR T cells into the tumors themselves.
I think we have two more online questions. One is your expectation that if this treatment were to work for MTC, you'd expect to see fairly immediate results, or would it take weeks or months? Because you're saying people could go back on TKIs pretty quickly.
I'm going to give a two-part answer. I think the hope is that there would be some kind of immediate response, and immediate is defined as, in this case, 2 months. So not the same as in leukemia, where it's almost instantaneous, but rather occurring relatively slowly over a period of a week to 2 months. I think if by 2 months there is no evidence of a decline in calcitonin, there's no evidence of any kind of tumor shrinkage or at least stabilization of disease, you would have to conclude that the CAR T cells are not working. Now, will the CAR T cells theoretically cause a complete remission within 2 months? Probably not, because of some of those challenges, physical challenges that I just spoke of in getting the CAR T cells into the tumors. The hope is that the CAR T cells will be catalytic, and what I mean by that is that even if there are low levels of them, they will survive, and they can keep doing their job very slowly over a period of time, chipping away, if you will, at these tumors in the body. And so that process might occur and take longer to happen over a period of weeks to months.
And one more question of, with the testing that was done on mice, is that confirmation CAR-T should be viable for treating solid tumors?
Yes and no. The mouse models are fake in the sense that cancer cells are grown up in the lab and then they are injected into the flank of a mouse. And in the flank of the mouse, they will form a tumor, which looks like a ball. But that tumor is not quite as sophisticated or complex a structure as a real human tumor. And I think this has been recognized for a century, that mouse models get you only so far. And so there is no natural mouse or monkey or any other animal model of medullary thyroid cancer. That would be ideally what you would test, and that simply doesn't exist.
Another question. is this study open to MTC patients of all mutations, or was it a limited? I can't remember what you said.
All mutations. So the most common question that I get is, what about RET? This is not a RET-directed therapy. So this particular therapy could be hypothetically effective in any kind of mutation, certainly not limited to RET.
I think that's all our questions, Dr. Cohen, and we are at the end of our time. Thank you
Perfect.
very much.
Yes, and I thank everybody for your attention. Thank you very much for the questions. They were terrific. I hope the answers were illuminating and will encourage interest in the trial. This is certainly a different way of treating medullary thyroid cancer, and I hope that we can continue to honor Dr. Barbos's memory and put his techniques to good use. Thank you very much.